Welcome back to What Did I Do in Lab today and last week, because I didn't do one for last week. I am sitting on my counter eating an entire bowl of KDs, so we love that for me. Here we go. So I didn't do one for last week because the only thing we really did was PCR, basic polymerase chain reaction. So all we did was take the DNA we extracted and purified from the week previous and load it up with all the chemicals and things and put it in the PCR in order to make a shit ton of copies of DNA, and that's not enough for a whole video. So today, what we did, we were running the gel of the DNA that we extracted, purified, and amplified. So we made our agarose gel, that is that cloudy thing up there. We had to just wait for it to solidify, do all that. Then we had a couple things we had to get done before. We had to add the running buffer to our DNA samples. We had to mix up our gel, wait for it to cool a bit, dump it into the thing, wait for it to fully solidify, take it out, add the buffer, all of that just to get it ready to run. Then when we were done, we ran our DNA samples in those little each one goes into an individual well. Those wells, if you can see that purple clip up there, you take that out, and where all those comb things are, those leaves indents into the gel, which you stick the DNA in, and the DNA is heavier than the buffer that it is in, so it sinks to the bottom. Then when all the DNA was loaded, we just put the lid on, and then just waited like 20 or so minutes for it to do its thing. You know it's working because you can see the bubbles going up the side. Then when it was all done, we took it out, stuck it in the machine, and got our result. So these were our results. This is admittedly not the best gel that I've ever done. It's also not the worst though. So in lane 1, we have our ladder that is just basically used to compare the different DNA fragment lengths. And then in lane 1, 2, and 3, or I guess only really 1, 2, those are my samples. So this is the DNA that I extracted from my own insect samples. And then there's supposed to be one in lane 3, but that one didn't really show up. I think I overflowed the well a little bit, so it got just overly diluted, so it didn't actually show up. And then the same thing happened in lane 5 and 7 for my lab partner's DNA. She also overfilled her wells a little bit because this was her first time doing that. I don't really have an excuse because I've done this probably like 20 times at this point, but... So ignoring the lane 4, 5, and 7, which don't actually have any DNA in it, this is our results. So this was testing for the presence of Wolbachia bacteria. And what we would see if there was Wolbachia present, there would be 2 distinct bands. Like in each lane, if there was Wolbachia, there would be a band up here, band down here. But in the 3 lanes that the DNA showed up in, there was only 1 distinct band. So basically what that means, in at least the 2 samples, or 3 samples that showed up well, there was no Wolbachia present in the samples. And yeah, that is the final of this lab. We will be starting a new one next week, so we'll be doing completely different things, but that is what we did today and last week.